Chapter 6, Anatomy of Flowering Plants. You can see very easily the structural similarities and variations in the external morphology of larger living organisms, both plants and animals. So, we can see, we can easily find out the similarities and differences. Variations means differences. So, similarities and differences in the external morphology means externally with the naked eye we can see the differences and the similarities in the external features of large living organisms like human beings, elephants, sharks, whales, or the angiospermic plants which are big in size. Okay, huge animals like the elephants we can see. Similarly, if we were to study the internal structure, one can also find several similarities and differences. Okay, internal structure also we can see many differences and similarities if we study about the internal structure. Okay, this chapter introduces you to the internal structure and functional organization of higher plants. Higher plants means angiosperms. Okay, the internal structure and functional organization. How functionally the plant is organized. Study of internal structure of plants is called anatomy. Okay, what is anatomy? Anatomy is the study of internal structure of plant is called as anatomy. Internally, how the cells are arranged into tissues and how the tissues what are the different types of tissues and what is the function of these type of tissues, what role they play. Okay, so that can be seen. Plants have cells as the basic unit. Cells are organized into tissues and in turn the tissues are organized into organs. So cell is the fundamental unit of life, the basic unit of life. Cells are organized into tissues. These tissues are organized into organs, and these different organs are they form the organ system. Okay, different organs in a plant show differences in their internal structure. Like in a human being, for example, how heart is different from lungs, lungs are different from the intestine is different from the stomach. Likewise, one organ is different from the other in plant body also. Leaves are different, stem is different, roots are different. Okay, within angiosperms the monocots and dicots are also seen to be anatomically different. Okay, so the monocots and dicots, they show internal variations. Okay, and internal structure also show adaptations to diverse environmental conditions. So the internal structure, it shows differences to according to the environmental conditions. Like you can see thick cuticle in xerophytes and thin cuticle in hydrophytes. The tissue system. So earlier we have discussed that. So the types of tissues based on the types of cells present. So if we consider how tissues varies depending on their location in the plant body, the structure and function would also depend on the location. Okay, so different tissues They vary in size and function from the location in the plant body. On the basis of their structure and location, there are three types of tissue systems. One is the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. So coming to the first type of tissue system, which is epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body. It forms the outermost covering. So as we all know, epidermis is the outermost layer, plant structure. So epidermal tissue system, it forms the outermost covering of the whole plant body, and it comprises of the epidermal cells, stomata, and other epidermal appendages. Okay, other linked things. So like the hairs and the trichomes and the hairs. Okay, the first tissue system is epidermal tissue system. It forms the outermost covering of the whole plant body. It has the components, the epidermal cells, stomata, which is majorly involved in the gaseous exchange, and the epidermal appendages. Appendages means other attachments like the trichomes and hairs. Trichomes are nothing but the extension of the epidermal cells, and hairs are the tiny structures. Okay, the epidermis is the outermost layer of primary plant body. Okay, the primary plant body, epidermis is the outermost layer. It is made up of elongated, compactly arranged cells. Compactly means there is no intercellular space between these cells. Okay, compactly arranged. They are very closely arranged. No intercellular space between the cells, which form a continuous layer. Okay. Epidermis is usually single layered, and the epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and with a large vacuole. Okay, the cell is parenchymatous with a very small amount of the cytoplasm and with a large vacuole. The outside of the epidermis is often covered with a thick waxy cuticle. So, okay, thick waxy layer, we call this layer as cuticle, which prevents the loss of water. The function of this cuticle is preventing the loss of water. Cuticle is absent in roots. All of us know, we know that roots are involved in the absorption of water. Hence, if the cuticle is present, they cannot absorb water from the soil. So, cuticle is absent in the roots. Okay. Next structure is stomata. These are present in the epidermis of leaves. Okay. The function of the stomata is gaseous exchange. Stomata, they regulate the process of transpiration and also the gaseous exchange. Okay, transpiration and also gaseous exchange. Each stomata is composed of two bean-shaped cells known as guard cells. Okay, these are dumbbell shaped in the monocots, bean shaped in the dicots, and with a small stomatal pore. You can see bean shaped structures, and in between you will have the stomatal pore. Okay. In grasses, the guard cells are dumbbelled. Okay. In dicots, it is bean shaped. In monocots, example grasses, the guard cells are dumbbell shaped. The outermost wall of guard cell are thin and the inner walls are highly thickened. Okay, you can see difference in the thickening. The outer wall is thin whereas the inner wall is thick. The guard cells have chloroplasts and also regulate the opening and closing of stomata. They have chloroplast and they regulate the opening and closing of stomata. So a few epidermal cells near the guard cells become specialized in shape and they are known as subsidiary cells. So the stomatal aperture, guard cells and the subsidiary cells put together we call it as stomatal apparatus. So these three components, the stomatal apparatus, guard cells and the subsidiary cells put together we call them as stomatal apparatus. So if we look at the structure of the stomata, so this is bean shaped, which can be seen in dicots. Inner thick walls, inner walls are thick, the outer walls are thin. So these are the epidermal cells, and surrounding the guard cells which are bean shaped, we have the subsidiary cells, and these subsidiary cells they have chloroplast, and inside these guard cells you can see the stomatal pore which will help in the gaseous exchange and also transpiration. So this is bean shaped stomata seen in dicots, whereas in monocots or grasses they are dumbbell shaped. Okay, dumbbell shaped. This is the epidermal cell and these are the subsidiary cells, and even these cells they have the guard cells, they have the outer thin wall and inner thick wall cells. Okay, and this is the shape difference. So please remember that the bean shaped stomata are guard cells are present in dicots, whereas the guard cells are dumbbell shaped in monocots. Okay, the cells of epidermis bear a number of hairs. Okay, the root hairs are unicellular. Root hairs are unicellular, and the elongation of epidermal cells, they help in the absorption of water. And nutrient minerals from the soil. Okay, and on the stem, the epidermal cells, the epidermal hairs, we call them as trichomes. On the stem, we call them as trichomes. If they are present on the roots, we call them as root hairs. Trichomes in the shoot system are multicellular. Please do remember, root hair is unicellular, whereas the trichomes are multicellular. They can be branched or unbranched, soft or stiff. They can be very soft, okay, or stiff, erect. They may even be secretory. The trichomes help in preventing water loss due to transpiration. They help in the preventing water loss due to the transpiration. Next tissue system is, we have the ground tissue system. After the epidermal tissue system, we have the ground tissue system. So all the tissues except the epidermis and the vascular bundles, they constitute the ground tissue system, except the epidermis and vascular bundles. Okay, except the epidermis and vascular bundles, they constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma and sclerenchyma. So parenchymatous cells are usually present in the cortex. So parenchymatous cells, you can see them in the cortex. Pericycle, pith and the medullary rays. Okay. So after epidermis, you'll have the epidermis, cortex, right? Cortex. In the cortex region, you can see the parenchymatous cells. So centrally, you will have the pericycle, and central pith, which gives rise to the medullary rays also. In the primary stems and roots. And in the leaves, the ground tissue consists of thin-walled chloroplast containing cells. We call them as mesophils. Okay, mesophils. You must have come across this mesophil cells in the photosynthesis. They aid in the photosynthesis process since they have the chloroplast, which consists of chlorophyll pigment. Okay, the next tissue system is vascular tissue system. So the name itself says that it deals with the vascular bundles. The vascular tissue system consists of complex tissues like the phloem and the xylem. Phloem and the xylem. Okay. The xylem and the phloem, they constitute the vascular bundles. We all know that xylem and phloem put together, they constitute the vascular bundle. In dicots stem, cambium is present between the phloem and the xylem. Such vascular bundle, because of the presence of a cambium, have the ability to form secondary xylem and phloem. So they are called as open vascular bundles. Okay, in dicots, in between the xylem and the phloem, we can see cambium. So such kind of vascular bundles, we are calling it as open vascular bundles. So xylem and also phloem, in between you can see this cambium. So cambium, so this phloem and xylem, it will form the secondary xylem or the wood, and secondary phloem it will form, and in between you can see the cambium. Because of the presence of cambium, we call this type of vascular bundles as open vascular bundles, which can be seen in the dicot stem. So in the dicot stem, you can see this type of vascular bundles. In monocots, vascular bundles have no cambium present in between. So if you look at this diagram, between the xylem and the phloem, there is no cambium in between the xylem and the phloem. Hence, they do not form any secondary tissues. So they don't form any secondary xylem or secondary phloem. So hence this type of vascular bundle is called as closed type of vascular bundle. Okay. If there is a cambium in between the xylem and the phloem, we call it as open type of vascular bundle, which helps in the secondary type of growth. Whereas if there is no cambium in between the xylem and phloem, that kind of vascular bundles are closed and it can be found in monocots. Okay. When xylem and phloem within a vascular bundle arranged in alternate manner with different radii, so such arrangement is called as radial arrangement. So this is the radial arrangement. Okay, this is arranged in, the xylem is arranged in one radius and phloem is arranged in another radius. This kind of arrangement we call it as radial arrangement, and it can be seen in roots. R for radial, R for roots. Okay, so we have another type, in conjoint type of vascular bundle. The xylem and the phloem are jointly situated along the same radius of the vascular bundle. So xylem and phloem, here these are situated along the same radius, and this is closed type because there is no cambium in between. This is open type, conjoint open type. Okay, such vascular bundles are common in stem and leaves. So you need to remember the type of vascular bundle and where it is found. That is very, very important. Okay, the conjoined vascular bundles usually have phloem located only on the outer side of the xylem. So xylem is always inside. Xylem is always inside. Okay, in exit, A-E-X-I-E-T. Exit, you have X, no? So X-I. So remember it that way, X-I. So xylem is always inside and phloem is always towards the outside. So here the most important thing is, so dicotyledonous stem, they have cambium. So if the cambium is present in between the vascular bundles, we call such type of vascular bundles as open type of vascular bundles. We can see it in dicot stems. Okay, if the cambium is absent, we call them as the closed type of vascular bundles. It can be seen in monocotyledons. Okay, so if the xylem and the phloem are present in an alternate manner along the different radii, we call such arrangement as radial type of vascular bundles, which can be seen in roots. R for radius, R for roots. Okay, remember it that way. And in conjoint type, xylem and phloem are jointly situated along the same radius. If it is different radius, we call such arrangement as radial arrangement, which can be seen in roots. And if it is same radius, we call such type as conjoint type. Example is, it can be seen in the stems and leaves. Okay, these are the different types of vascular arrangements which can be seen in the vascular tissue system. Okay, we have read about the epidermal tissue system. Okay, epidermal tissue system, ground tissue system, and vascular tissue system. Here you need to remember the types of vascular bundles along with their examples. Next, we have the anatomy of dicot and monocot plants. So till now we have seen different tissue systems, the epidermal tissue system, the vascular tissue system, and also we have seen the ground tissue system. So to get a much more clear picture, so we should get into the much more details. For a better understanding of the tissues, organization of roots, stem and leaves, so it is convenient to study the transverse section. So if we cut open the plant transversely, means horizontally, so we can find the matured zones of these organs, that is roots, stem and leaves. So it will give a much more better clear picture of the tissues of the plant. So first we'll be talking about the root. In that we have the dicot root, dicotyledonous root. Okay, let us look at the dicot root. internal structure transversely how it is. So the transverse section of sunflower root, here we have taken the example of sunflower root. It is as follows. It has the outermost layer is epiblema. It has the outermost layer which we call it as epiblema, and many cells of the epiblema, they come out or protrude in the form of unicellular hairs. Okay, unicellular hairs. Here you can see hairs, root hairs. These are unicellular. Okay, and they have originated from a layer known as epiblema. The cortex consists of several layers of thin-walled parenchyma cells. Cortex is made up of thin-walled parenchyma cells, okay, with intercellular spaces. With the intercellular spaces. So you have the epidermis, which is the outermost layer. After the epidermis, you will have the cortex. See, in the epidermis, there is no intercellular spaces. It is made up of the parenchymatous cells. Okay, whereas the layer after the epidermis, you will have the cortex, which has intercellular spaces in between. Okay, it is thin wall. The innermost layer of the cortex is endodermis. Innermost layer, we call it as endodermis. The innermost layer of the cortex, we are calling it as endodermis. This is the endodermis. Okay, it comprises of a single layered barrel shaped cells without any intercellular spaces. So endodermis is single layered and the cells are barrel shaped. Okay, this is the most important point which you need to remember. The innermost layer of the cortex, we call it as endodermis, and the cells are barrel shaped without any intercellular spaces. The tangential as well as the radial walls of the endodermis have a deposition of water impermeable waxy layer which is suberin and in the form of Casparian thickenings. Okay, it is impermeable to the water. It will not allow the passage of water. This is these Casparian strips. It is the most important point. Please do remember. Casparian strips can be found in the internal structure of the root, dicot root, along the tangential as well as the radial walls. So this endodermis, how many layered it is? It is one layered without any intercellular spaces. This Casparian strips is made up of suberin and it will not allow the water to pass through them. So next to the endodermis, you will have a layer thick-walled parenchymatous. Thick-walled parenchymatous layer, we call it as pericycle. Pericycle. Inner to the endodermis, you will have the pericycle. This is the pericycle. So the initiation of lateral roots. Most important thing, what is the function of pericycle is lateral roots and vascular cambium. The vascular cambium during the secondary growth takes place in these cells. Okay, the lateral roots and also the vascular bundle is formed from the pericycle which is present next to the endodermis. Okay, next is you will have after the pericycle you have the pith. Pith is the centralmost part. Okay, this is the pith. The pith is small or inconspicuous, so it is almost unseen, and the parenchymatous cells which lie between the xylem and phloem are conjunctive tissue. Okay, this is the conjunctive tissue. There are usually 2 or 4 xylem and phloem patches, and a cambium develops later on. Cambium will develop in between these vascular tissues during the secondary growth. All tissues on the inner side of the endodermis, such as pericycle, vascular bundle and pith, they constitute the stele. Okay. So the pericycle, vascular bundle and the pith, these three components, they constitute the stele. Okay, stele, central stele. The pericycle, after the endodermis, whatever is there, the pericycle, okay, the vascular bundles, these are the vascular bundles, and the central pith, these three components, they constitute the stele. Okay, this is about the structure of dicot root. Okay, it has the outer layer which is epiblema, from which root hairs will occur. After that you will have the epidermis, which is made up of parenchymatous cells without any intercellular spaces. After that you have the cortex, which are loosely arranged with some intercellular spaces. The innermost layer of cortex, we call it as endodermis. So the radial and tangential walls of the endodermis has suberant depositions. So these depositions, we call it as Casparian strips. So next to the endodermis, you will have the pericycle, which are important in the formation of lateral roots, and vascular cambium during the secondary growth. After that you will have the central part, which is called as pith. Okay, and in between you will have the xylem and phloem. So xylem and phloem, and during the secondary growth, in between the xylem and phloem, you can see the formation of cambium. Okay, so the tissue after the endodermis forms the stele. So what constitutes the stele? That is the pericycle, vascular bundle and the pith. These three components, they form the stele. PVP. Okay, PVP pipes. Okay, they constitute the stele. The pipe is made up of steel. In that way you can remember. This is about the internal structure of dicot root. Next, coming to the monocot root. Okay, the anatomy of monocot root is somewhat similar to the dicot root. It has an epidermis like the dicot root. It is also having epidermis, cortex. So after the epidermis, you have the cortex, and the innermost layer of cortex we have called it as endodermis. And here also they have the pericycle and vascular bundle and pith. Okay, the components are almost similar compared to the dicot root. So this is the outermost layer, epidermis. From the epidermis you can see the extensions, which are the root heads. The epidermis has a very compactly arranged parenchymatous cells without any intercellular spaces. Okay, as compared to the dicot root, which have fewer xylem bundles. So once we'll see. So epidermis, below this cortex. In cortex you can see tiny intercellular spaces like the dicot root. Okay, below the cortex you can see the innermost layer is endodermis. Okay, endodermis, and below the endodermis you will have the pericycle. Okay, now let us see what is the difference as compared to the dicot root, which have fewer xylem bundles, which are usually more than 6. In monocot root, you will have more number of xylem and phloem bundles. So here you can see usually 6 xylem bundles. Such condition we call it as polyarch. Polyarch condition can be seen in monocotyledons, monocot root. Okay, polyarch condition. And pith is large and well developed. The pith is large, whereas in the dicot root, the pith is very small or inconspicuous. So in the monocot root, the pith is very large. And also monocot roots do not undergo any secondary growth. Hence, here there is no cambium. There is no cambium. Whereas here you can see the lateral roots and also the cambium. Cambium and lateral roots growth can be seen, which is given by the pericycle. Whereas hence in the monocot root there is no secondary growth. You cannot see any cambium here. Okay. Yeah, this is the major difference between the dicot root and the monocot root. So coming next to the structure of dicot stem. The transverse section of a typical young dicotyledonous stem shows epidermis as the outermost protective layer. Here also the outermost protective layer is stem and it is covered by a thin layer of cuticle. So the stem is covered by a thin layer of cuticle. In roots, we have seen the root hairs. Okay, the extension of the epidermis is root hairs. Since the roots are they participate in the process of absorption of water. Root hairs help in the absorption of water and minerals from the soil. Here the stem, the outermost layer is epidermis and it is covered with a thick layer of cuticle. In the dicot stem, okay, it may bear trichomes and few stomata. Okay, the cells are arranged in multiple layers between the epidermis and pericycle and they constitute the cortex. Okay, likewise same, and the cortex consists of three subzones. The cortex, it consists of three subzones. Outermost layer is hypodermis. It is made up of few collenchymatous cells. Collenchymatous cells. Hypodermis is made up of collenchymatous cells. And just below the hypodermis, you will have the cortical layer. And the function of this hypodermis is providing the mechanical strength to the young stem. When the stem is in young condition, this collenchyma will provide the stem with the mechanical strength. Means it will not get broken very easily. If you can see the green stems, it is very difficult to break the green stem. Okay, it is because of the collenchymatous cells. Next is the cortical layer below the hypodermis. It has a rounded thin-walled parenchymatous cells. So it will have rounded thin-walled parenchymatous cells with conspicuous intercellular spaces. So you can see clear intercellular spaces. And below this cortical layer, you will have the innermost layer made up of again the endodermis, which is rich in starch grains. Hence, this layer is known as starch sheet. Okay. So, in the dicot stem, the cortex is divided into three zones. Hypodermis, which is made up of the collenchymatous cells, which help in the giving the mechanical strength to the young stem. And below this, you will have the cortical layer, which is made up of parenchymatous cells with very much intercellular spaces, huge intercellular spaces. And the lowermost or the innermost layer, we call it as endodermis, which has this starch grain, which is the food material. Hence, we call this innermost layer or the endodermal layer as the starch sheet. Starch sheet. So, if you can see here, if you can see this, so the outermost layer, this is the epidermis. Okay. So epidermis, below this you will have the cortex. The outermost layer of cortex is made up of collenchymatous cells, which helps in the giving the mechanical strength to the young stem. Below this, the parenchymatous cells with the intercellular spaces, and innermost layer is the endodermis. Okay. So this is the transverse section, this is the radial section. So both the cells. So inner to the endodermis you will have the pericycle. Pericycle, let us see the function of pericycle here. The pericycle present on the inner side of the endodermis and above the phloem in the form of semilunar patches is made up of sclerenchyma. The pericycle is present in the form of semilunar patches. Okay, semilunar means half moon shape. This is half moon shape and it is made up of sclerenchyma. Okay, and in between the vascular bundles there are few layers of radially placed parenchymatous cells which constitute the medullary rays. So radially placed parenchymatous cells, they constitute the medullary rays. These are the medullary rays, if you can see this. So medullary rays. And you have a large number of vascular bundles are arranged in a ring arrangement. The ring arrangement of vascular bundle is the characteristic of dicotstem. Very important point. A ring arrangement, ring type of arrangement. The vascular bundles are arranged in the form of a ring. Okay, ring type of arrangement of vascular bundles is the characteristic feature of a dicot stem. Okay, and each vascular bundle is conjoined, open with endark protoxylem. Means conjoined means the xylem and phloem are present on the same radius. Open means we can see cambium between the xylem and the phloem, and endark means the protoxylem is towards the pith and the metaxylem is towards the outside. Okay, so such kind of protoxylem we call it as endark. This endark condition can be seen in the dicot stem. Okay, very important point. Ring arrangement of vascular bundles and conjoint, open, endark protoxylem can be seen in dicot stem. A large number of rounded parenchymatous cells with large intercellular spaces occupy this central portion of the stem. They constitute the pith. So in the dicot root also, so you must have seen the dicot root, the pith is inconspicuous, whereas here the pith is very much central portion, it is very much conspicuous. Okay, they occupy the central portion. Okay, next we have the monocot stem. Coming to the monocot stem. So the monocot stem has a sclerenchymatous hypodermis. So here also the hypodermis is made up of sclerenchyma, and it has a large number of scattered vascular bundles. The vascular bundles are not arranged in the form of a ring, rather the vascular bundles are scattered and each is surrounded by sclerenchymatous bundle sheath. Large conspicuous parenchymatous ground tissue, and the vascular bundles are conjoined and closed. So they are present on the same radius and it is closed. The peripheral vascular bundles are generally smaller in comparison to the centrally located ones. Okay, the vascular bundles which are present towards the sides are smaller in size. Peripheral one are very smaller in size in comparison with the central vascular bundles. The phloem parenchyma is absent. Phloem parenchyma is absent. And water containing cavities are present within the vascular bundles. Okay, just once look at the diagram. So the monocot, it has the outermost layer epidermis and the hypodermis is made up of sclerenchyma. Okay, so it is here the cortex is not divided into different zones like the dicot plant. Okay, the vascular bundles are scattered. The vascular bundles are scattered. They are not arranged in a radial way like the dicot stem. Okay, here the xylem and phloem are present, and between you have the ground tissue. Ground tissue can be seen here. Okay, so this is about the dicot stem. So we have the dicot leaf, which is also known as the dorsiventral leaf, because we can differentiate the dorsal surface and the ventral surface in dicots. Okay, the vertical section of a dorsiventral leaf through the lamina shows three main parts, namely the epidermis, mesophyll, and the vascular system. Okay, it will have the epidermis. So the epidermis consists of the upper epidermis, which is the adaxial epidermis, adjacent to the axis, means adjacent to the stem axis, and the lower epidermis we call it as abaxial epidermis. Abaxial epidermis means the lower epidermis, back of the stem. Okay, back of the stem. So adjacent to the axis is adaxial surface or the upper surface, and opposite to it you will have the lower surface or abaxial surface, and the leaf has a conspicuous cuticle. The dicot leaf, it has a conspicuous cuticle. The abaxial epidermis or the lower epidermis, it will generally bear more stomata. So remember, more stomata are present on the lower surface of the leaf in comparison with the upper epidermis and then the adaxial epidermis. The later may even lack stomata, means sometimes on the upper epidermis the number of stomata may be absent completely. The tissue between the upper epidermis and the lower epidermis, we call it as mesophyll. The tissue which is present between the upper and the lower epidermis, we call it as mesophyll, and these mesophyll, they possess the chloroplast. So they help in a very important process, which is the photosynthesis, and it is made up of two types of parenchyma. One is the palisade parenchyma and the spongy parenchyma. So adaxially present palisade parenchyma is elongated and arranged parallel to each other. Means the upper epidermis has the palisade parenchyma. They are elongated and they are arranged vertically and parallel to each other. Okay, the oval or round and loosely arranged spongy parenchyma is present below the palisade cells, and they extend to the lower epidermis. Okay, so there are numerous large spaces and air cavities between these cells. Okay, next component we have the vascular system. The vascular system can be seen in the veins and the midrib. The leaf, the veins and the midrib, they have the vascular system. The size of the vascular bundle is dependent on the size of the veins. The veins thickness varies. The reticulate venation of the dicot leaves. The vascular bundle are surrounded by a thick wall layer known as the bundle sheath cells. Okay, they are surrounded by a thick wall cell known as bundle sheath cells. Okay, now just we'll see the diagram once. So this is the adaxial epidermis or the uppermost epidermis, and this is abaxial epidermis or the lower epidermis. So in between the upper epidermis and the lower epidermis, you will have the parenchymatous cells, mesophyll cells with the chlorophyll pigment. Okay, chloroplast, it has chloroplast which has the chlorophyll pigment, and this pigment will help in the process of photosynthesis. So it has two types of parenchyma cells. One is the palisade parenchyma cells, which are elongated, vertically elongated, and you have the spongy parenchyma cells. So with intercellular spaces, this spongy mesophyll cells, it has intercellular spaces, and I told you the lower surface will have stomata, and this opening of stomata, so this lower epidermis, it has stomata and an opening which we call it as stoma. It will lead into substomatal cavity. Okay, so it also has the air cavities and the vascular bundles. So it will have the xylem, and phloem, and surrounding these vascular bundles will have a sheet which we call it as bundle sheets. So since they encircle the vascular bundles, this sheet we call it as bundle sheet. The bundle sheet which covers the vascular bundles. So this is about the structure of dicot leaf. Lastly, we will see the structure of the monocot leaf. The monocot leaf is also known as isobilateral leaf, means it is similar on the both the sides. The anatomy of isobilateral leaf is similar to that of dorsiventral leaf, which is nothing but the dicot leaf, in many ways. It shows the following differences. So we shall see the what are the differences. The isobilateral leaf, the stomata are present on both the sides, whereas in the dicot leaf, we have seen the stomata are present mostly on the lower surface only, whereas in the monocot leaf we can see stomata are present on both the surface of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. Okay, in dicot leaf the mesophyll is differentiated into palisade parenchyma and spongy parenchyma. The palisade parenchyma cells are elongated cells. Okay, they are elongated vertically, and you have the spongy parenchyma also with intercellular spaces in between them. Here there is no such differentiation in the mesophyll. Okay, in grasses, certain adaxial epidermal cells, means the upper epidermal cells along the veins, they modify themselves into large empty colorless cells. Such cells we call it as bulliform cells. So these bulliform cells are colorless. So in color spelling you have O, bulliform cells also you have O. So they are colorless, without any color, colorless, zero means without any color, remember it that way. When the bulliform cells in the leaves have absorbed the water and turgid, the leaf is exposed, and when they are flaccid is in condition, means when they lose the water, so the leaves curl inward to minimize the water loss. So the curling and uncurling of the leaves in monocots is because of the bulliform cells. These cells are colorless cells, large empty colorless cells. Okay, and the parallel venation, in this you can see the parallel venation, whereas in dicots mostly you can see the reticulate venation. Okay, the parallel venation in monocots is reflected in the near similar size of the vascular bundles, whereas you can see difference in the size of the vascular bundle in the dicot leaf. Okay, except in the main veins. Okay, so these are the main differences between the monocot leaf and the dicot leaves. So this is the adaxial epidermis, abaxial, upper epidermis and the lower epidermis. This is also having the stomatal cavity, which leads into substomatal cavities. Okay, and there is no differentiation into mesophyll and the palisade layer here. The mesophyll tissue is made up of only one type of cell. Okay, only the you can see only one kind of cell. Okay, so and also the xylem and the vascular bundles, they are of almost same size. So this is about the monocot leaf. Most important thing here is they have the bulliform cells, which are colorless cells. They help in the rolling and unrolling of the leaf. Okay, thank you.